Good afternoon, everyone. So today we are going to start our new chapter. All right. So before starting our new chapter and today's class, so I'm launching a technical poll so that I get to know whether I'm clearly audible and visible to you or not. Okay. So here is the poll. You can just give me a response. Okay. So here I'm asking the poll that I am clearly audible and visible to you. So now we are going to start our today's new chapter, that is cell and the unit of life. Okay, so as we know, we have done previous chapters. Today we are going to cover the cell unit itself, right? So we are let's start the class with the recall test. So your first question for the today's class is select the wrong statement among the given four statements. So your first statement is bacterial cell wall is made up of peptidoglycans. B. Villi and fimbriae are mainly involved in motility of the bacterial cell. C. The cyanobacteria lack flagellated cells. And D. That mycoplasma is a wall-less microorganism. Okay, so you are having 30 seconds. In 30 seconds, you have to give your response. For that, I am launching the poll. Okay, so your 30 seconds has already started. So here I'm launching the MCQ poll. Okay, so within 30 seconds, this poll will get end up. Right, so you have to find out the wrong statement among the given statements. We are having four statements which are saying that one is saying bacterial cell wall is made up of peptidoglycan. Second is saying villi and fimbriae are mainly involved in motility of bacterial cell. C is saying cyanobacteria lacks flagellated cells, and D is saying mycoplasma is a wall-less microorganism. Okay, so here we are about to complete 30 seconds. So with this, I'm ending the poll. Okay, so here you can see the result that most of you have answered option B. Okay, so let's check what is the correct answer. For that, we are going to discuss each and every statement. So, the A statement is saying bacteria cell wall is made up of peptidoglycan. Yes, we have seen that bacteria, which we have seen, they are having the cell wall, which is made up of NAG and NAM, right? NAG and NAM, which is a peptidoglycan. Then options B is saying that fimbriae and the are mainly involved in the motility. Motility means movement, that is locomotion. But if we see that fimbriae and the villi, these are the two different projections which are present on the surface of the body of a bacteria. Villi are very small in number, right? They are very few in number, and it is a very rigid structure, like a tunnel, like a tube, which is projecting out of the cell, and it is only present when there is F plus plasmid is present in a bacteria, right? So due to the presence of this F plus plasmid, this villi is present and leading to the process called conjugation, right? So yes, right. So we can say the villi is not at all involved in the locomotion. Next, we are having the fimbriae. Fimbriae is again small hair-like structures which are present on the surface of the bacteria, right? And if we see this structure, this fimbriae are helping in the connection. They are helping in the connection of the bacteria with the solid, right? With the solid support. Okay. So with this, we can say the villi and fimbriae, these both structures are not at all involved in the motility, but instead they are involved in the process of conjugation, that is transfer of DNA, and involved in the contact with the solid support, right? Next statement is saying cyanobacteria lacks flagellated cell. Yes, because here we can include the example of Anabena, which is a blue-green algae, which is a filamentous structure having a mucilaginous covering on it and helping in the nitrogen fixation, right? So it is not having flagella. Then mycoplasma are the smallest among bacteria. They are the smallest size of bacteria which are not having any cell wall and leading to the pathanogenicity, right? So as we have studied all the four statements, we have found that option B is the correct one, right? So on that basis, we can say that statement B is our correct answer. Okay. So now moving to the next recall test are, so next test is, and next question is a match the following question, right? Here we are given the two different columns. In one column we are given some structures, and in column two there is an explanation regarding that particular structure. Okay, so what you have to do, you have to just look at the options and match which description is given for a particular structure, right? So for that I'm giving you next 35 minutes. Okay, for next 35 minutes I'm launching the poll. 35 seconds, sorry. So in 35 seconds you have to match it and give the answer, and give your poll inside this MCQ poll. Okay, you have to feed your answer. So here I'm sharing the MCQ poll. Now you are having 30 seconds, because already we have talked about 30 seconds to read the question properly. Along with that, I am reading the questions for you. That option A is, the A is having thylakoid, B is having cisternae, C cisternae, and D is chromatid. Okay, and in the column 2, as you can see, four statements are given describing the structure of these four structure further, right? So okay, I think some technical issue was there due to which you are unable to give your response. So I have launched the poll again for next 15 seconds. Kindly give your answer. Okay, so here I'm ending the poll. As you can see, the 100% result came for the option D, right? So let's see the option D is correct or not. Okay. So first, we know that whenever we have to do such kind of question, we always go through the options, right? So here, as we can see, out of A, B, C, D, the B option, for the B option, we are having good variety, right? So here we can see B is given for cristae, right? As the name itself is very small, so cristae are the foldings of the inner mitochondrial membrane, right? So we have to see where these foldings are given. So when we see the infoldings of the mitochondria are given by 4. So when we see here again in options, so for B, the fourth statement is given by the two options, that is option B and D. So we can eliminate the option A and C on the basis of that. Now we have to find out which option is having the different statement. So here we can see by choosing the C, we can distinguish which option is actually correct. So here the C option is the C uh, term given is cisternae. And as we all know, cisternae are the disc-like structures. These are the long foldings of the vessels which are forming the Golgi body, right? So if we see it for C, it is belonging to the statement 1, which is given in column 2. So accordingly, if we see, so for that we are matching with the option D. So yes, the option given by you is the correct answer. Okay. So now we are moving towards our today's chapter, and today we are going to start with the cell chapter. So as everyone knows, the cell is the fundamental unit of life. Fundamental, what is the meaning of fundamental? That is, it is making a structural and the functional unit of life. Structural, as you can see, whenever we go to the making of house, the house can be built only with the help of bricks. In the very same way, when an organism body is built, the single structure which is required for the building is a cell. Okay. And we know a single cell is capable of performing all the kind of uh, uh, activities. So yes, the cell is a structural and functional unit of the life for all living organisms. With this, we are moving toward the depth of this chapter, right? So as we know, the Robert Hooke was the first person. He was the first scientist who has discovered the cell by the use of his own made microscope, right? He made his own microscope, and in his own microscope, he has seen a thin slice of a cork. He has seen a thin slice of a cork, and when he observed that thin slice of a cork under the microscope, he saw some honeycomb-like structure, right? Some honeycomb-like structure which are having only cell wall, right? So these honeycomb-like compartment or the structure which he see in the thin slice of the cork, he called them as a cell, right? So what the cell observed by the Robert Hooke was a dead cell initially, right? So after the Robert Hooke, the next invention or the discovery was done by the Anton von Leeuwenhoek, who was the first scientist who discovered the first living cell by using the sample of water, right? He observed the stagnant water under the microscope, and there he saw like some small microscopic units are moving throughout that water. So he was the first person who discovered the living cell. Okay. Is it clear to you? If it is clear to everyone, please give me a thumbs up. Okay, then. So let's move further. So as we have know about the cell, some specific theories are given. The first theory, which is came for the cell, is given by the Shieldin and the Schwann. Shieldin and Schwann, these are the two different person and done their discoveries in two different regions, right? So one is botanist, one is zoologist, and both of them worked on the different components, but at the same time, they explain all the same things, and when we combine their proportions, their postulates, we lead to the formation of the cell theory. Okay, so that is how the cell theory was formulated. And when they have observed the plant and animal cell, what postulates they have given, that all the living organisms are composed of cell and their product. This is the initial statement given by the Sheridan and Schwann, like cell is the fundamental unit of life, and second, that each and every organism is composed of cell. Okay, but when the Rudolf Virchow came in life in 1855, he done the next explanation. He has expanded the cell theory. See, mind this thing, that the cell theory was proposed by Sheldon and Schwann, but it was expanded by the Rudolf Virchow by adding one new statement, that is saying each and every cell arises from the pre-existing cell, that is omnis cellula a cellule. Okay, so with this third statement, we have expanded our cell theory and leads to the final cell theory, which is saying, again, I am repeating, the cell is the fundamental unit of life. Each and every organism's body is made up of cell. And the third one is the cells arise from the pre-existing cell. Okay, is this clear to everyone? Is it clear? Okay. So let's move to the next topic, that is prokaryotic cell. Initially, we have seen the cells. The cells can be distinguished into two different forms. One is prokaryotic, another is eukaryotic. Can anyone tell me on what basis we have distinguished these two cells, or these two types of cells we have categorized? On what basis? You can give your answer in chat box. Yes, that is presence of nuclear membrane. Very good. Yes, that is due to the presence. Very good, Gopal. Yes, that is correct. On the basis of the presence of true nucleus, right? So we have seen in prokaryotic cell, the cells are having genomic material, but that genomic material is not at all enveloped or packed in an envelope, which we usually call the nuclear membrane, right? So their genetic material was floating in the cytoplasm freely. So we were not having any proper structure for the nucleus. And the structure of the nucleus without the nuclear membrane is known as nucleoid, right? So that is how we have distinguished the prokaryotes and eukaryotes. So if we see the term itself, prokaryote, that means pro, primitive. Eukaryote, nucleus, or we can say the false nucleus. When we see the term eukaryotes, eu means true or definite, and karyotes means nucleus, right? Okay. See, DNA which is present in a prokaryotic cell, we are also having some extra double-stranded circular structures, right? Those are known as plasmids, right? So these plasmids are not at all a genetic material, because whatever the morphology of a cell we are seeing, that is provided by the DNA, which is forming chromosome, right? Which is present in a nucleus, right? But the plasmid are providing extra help. They are providing extra help and features to the bacteria. So that is why we are not calling plasmid a genetic material. Okay. Yeah. Okay then. So as we can see, this prokaryotic cell, basically we include here bacteria in it, right? So when we see the structure of bacteria, what we can see, the bacteria is having a cell envelope, right? The envelope is itself composed of three different layers. The innermost is plasma membrane, the middle one is cell wall, and the outermost is the glycocalyx. Glycocalyx is a mucilaginous. It is a mucilaginous covering which is present outside the surface of bacteria. Okay, so all these three structures are making the cell envelope of a bacteria, right? And when we combine this glycocalyx and cell wall together, we lead to the differentiation of the bacterial cell as the gram positive and the gram negative. If the glycocalyx is thick, right, it is impossible for the bacteria to retain the stain, so they become glycocalyx. So depending on the thickening of the glycocalyx, we have distinguished bacteria as gram positive and gram negative. If thickening is there, if thickening is present, they can retain, they cannot retain the stain. But if thickening is absent, right, they can no. So on that basis, we can say yes, the glycocalyx is helping in the differentiation of gram positive and negative bacteria. Okay. Next, we are moving towards the mesosomes and the chromatophores. As we all know, the prokaryotes are the organisms which are not having the cell-walled organelles, right? They are not having the membrane-bound organelle, but still the cell is performing all the functions like photophosphorylation, like the oxidative phosphorylation. Oxidative phosphorylation, can anyone tell me where the oxidative phosphorylation is used? Where this oxidative phosphorylation is used? Or what is the meaning of this oxidative phosphorylation? Yes. So, yeah, this is I'm going to explain. So, oxidative phosphorylation, that is the formation of ATP, right? And this process is usually done in mitochondria, but as mitochondria is not present because it is a membrane bound organelle, so all this oxidative phosphorylation has to be performed in a particular region. So as you can see here in the bacterial cell, there is an inclusion, there is an inclusion body, right? You can see here some enfolding of a plasma membrane is present. This enfolding of the plasma membrane is providing a cavity which is known as mesosome, and mesosome is a site in the prokaryotic organism where this oxidative phosphorylation is taking place. Yes, we can say the mesosomes are acting as an analogous to mitochondria because all the oxidative phosphorylation is taking place in mitochondria. Okay. So next we are moving towards the chromatophores, right? These chromatophores are the pigments. They are the pigments which are found in the bacteria. They can be of different types, right? And if you see the cyanobacteria, which we also call as blue-green algae, they are having like chlorophyll, right? Which is helping in the photosynthesis. So these chromatophores, these are the colorful pigments which are present in a bacterial cell and having a particular function. Next, we are moving towards, so as I have already explained you the genomic DNA and the plasmid DNA. The genomic DNA is a double linear DNA, right? Single chromosome genetic material. It is naked because it is not having a covering of membrane. But if we look at the plasmid, it is a double-stranded circular DNA, which is a smaller in size and present in the cytoplasm, right? Separate from the genetic DNA. Okay. Next, we are moving towards the bodies which are present on the surface of the cell. First is flagella, right? As you can see here, two different kind of structures are protruding out of the bacterial cell. One is a long hair-like structure and very small green color, small hair-like structures which are present on the surface of the bacteria, right? So if we saw about this, let's talk about the first long hair-like structure which are present in a very few amount. These are known as flagella. Okay, and the flagellum is itself composed of three different components: basal body, hook, and the filament. The basal body is providing the attachment of the flagella to the structure of the bacteria, from where it get the motion or it get the bidirectional rotatory motion, due to which flagella will do this kind of movement and helps in the locomotion of the bacteria. Okay, they are protruding out of the surface of the bacteria. Yes, the flagellas are having 9+2 arrangement because they are made up of centriole, right? You will see in the uh, next slides that centriole is present in the arrangement of 9+2. So here we are having doublet, 9 doublets. 8 doublets are present in the periphery and 2 doublet, and 1 doublet is present in the center of the arrangement. Okay, that I will explain you in the upcoming slides. Okay, so filament is the longest portion protruding out of the body and providing the locomotion to the cell. Along with that, the body is also covered with the cilia. That cilia is also acting as a locomotory organ on the body of the prokaryotic cell, right? So next we are moving towards the next other two types of structures. One is fimbriae and another is sex phylus. So as you can see, fimbriae is just looking like similar to the cilia, but there is difference in the function of the cilia and the fimbriae. As I told you, cilia was responsible for the locomotion, whereas fimbriae are used for the attachment of the bacteria with the solid support. Okay, so here you can see the bacteria, they help to attach the rock in the stream and to the host tissue, right? And I have also explained you that phylus are due to the presence of F plasmid, right? F plasmid, that is fertility plasmid. If the bacteria is having F plus plasmid, that is fertility plasmid is present in that bacteria, only then this phylus structure is present, and this phylus structure helps in the transfer of DNA. Okay, it helps in the transfer of DNA by the making a phylus bridge, and that process of transfer of DNA by the making of phylus bridge, that is known as conjugation. Okay, that is known as conjugation. There I can explain it to you. So this is one bacteria. This is F having F plus plasmid. So it is having a bridge-like structure. Yeah, bacteria can also reproduce by sexual means. That sexual mean is actually the transfer of the DNA. Okay, so here I changed the color of my pen so that you can see it clearly. So this blue bacteria is one bacteria, and another bacteria we are having the yellow one, right? So both are having F plus plasmid, but it is difficult to distinguish which one is male and which one is female. When you see this bacteria, they are having DNA as their genetic material. But this genetic material, we are not talking about here, we are talking about the plasmid DNA, right? So what happens? By the help of this bridge, they usually transfer their DNA from one to another organism. So this exchange of DNA is actually the means of sexual reproduction in bacteria, explained as conjugation. Okay. Okay, so next structure which is present in the prokaryotic cell, which is a non-membranous structure, non-membranous, and the smallest structure, non-membranous, and the smallest organelle is ribosome, which is denoted by 870S, because we are talking about the prokaryote, so it is denoted by 70S. This S in 70S, it actually denotes the Sedberg unit. That means it is defining the sedimentation rate of the ribosome. As we say, it is the smallest component, so it diffuses, it's settled at a very high speed. Okay, so why 70S? Because it is composed of two different units. One is 30S unit, which is the smallest unit, and another is a 50S unit, that is a largest unit. But when we talk about the same ribosome in the eukaryotic cell, the are having 80S ribosome, which is composed of as 60S and 40S unit. 60S unit is a bigger one, and 40S unit is a smaller one. Okay. Okay, they get attached by the reactions, right? Whenever they have to form the protein, which we study in the translation in 12th class chapter, we have studied. Okay, so whenever the translation has to happen, the mRNA, here I'll make the mRNA stands on the surface of this smaller subunit, right? Because it is acting as a platform. Over that, the bigger unit came and overlapped. So here you can see the mRNA, the RNA get sandwiched in between these two subunits, and That is how these two units helping in the translation of this RNA and leads to the formation of protein. Okay, that you will study in the translation unit properly. Okay, so you got about the ribosome. Ribosome is a protein making factory. It is a protein making factory. Okay, next we are having inclusion body. Some another crystal-like structures which are present inside the body of a bacteria. Okay, if we see this structure, these are again non-membranous structures. These are non-membranous structures because these are the storage capsules, right? Or we can say these are the reserved material which get aggregated and forming a granular. Okay, so they can be found freely floating in the cytoplasm of the bacteria, and these granules can be composed of inorganic salts of phosphate and sulfate. They can be made up of carbohydrates. They can be a cluster or collection of the gas, right? So these are providing the analogous function of a vacuole here, right? In eukaryotes, the vacuole are serving as a membranous sac for the storage of all the components. In the very same way, in bacteria, or we can say the prokaryotic cell, we are having the inclusion body working for the storage of stuff. Okay, next we are having moving towards the eukaryotic cell. That means the cell which is having well-defined nucleus as well as the membrane-bound organs. Okay, so these are the components which are present in the plant cell, because eukaryotic cell we can distinguish as a plant cell or as an animal cell, right? If the eukaryotic cell is having a cell wall, if a eukaryotic cell is having a cell wall, it is considered as plant cell, whereas the another cell which is not having the cell wall, they are known as the animal cell. Okay, so these are the organelles which are found in the plant cell. The cell membrane, which is common for both the plant cell and the animal cell because it is a plasma membrane. The external boundaries, which is extra present in a plant cell, that is cell wall, which is made up of cellulose, right? Which is made up of cellulose. Then endoplasmic reticular system, that is we call ER, right? ER can be of two types, smooth and rough. We will discuss it later. Then in this endomembrane system, we are having Golgi bodies, lysosomes, and vacuoles. Why we are calling all the group of all these four organelles as an endomembrane system? Because these organelles are involved in the formation of membrane, right? They together work for the formation of membrane, so we call them as endomembrane system. Next, we are having mitochondria, a ATP producing site, right? The plastids, which are colorful, which can be colorful or colorless. Pigments, which are helping in the storage as well as photosynthesis in a plant. These are only present in plants. Okay, they cannot be found in animals. Then the ribosomes. What kind of ribosome we are having in eukaryote? 80S or 70S? Yes, that is 80S. Next is the cytoskeleton. As you know, the cell is having a specific shape, right? So just like our body is having proper shape because of our skeleton. If we are not having skeleton, our body will be behaving like a dough, right? So to provide a specific shape to the body, we require skeleton. In the very same way, the cells are also having skeleton, which is a machine network present beneath this membrane, the plasma membrane, providing a supporting structure to the cell and the plasma membrane. Next, the cilia and the flagella, which we have already discussed in prokaryotic cell, the centrosome and the centriole. These are the extra stuff, right? But these are not present in plant cells. They are present in the animal cell. They are working during the division of the cell. Next, we are having the nucleus, which is also known as the control center of the cell. And last, we are having the microbodies, because these microbodies are required in the cleaning of the cell. Microbodies, just like we are having peroxisome and glyoxysome. The peroxisome is helping in detoxification of hydrogen peroxide molecules, whereas glyoxysomes are required whenever the carbohydrate content become low in a cell, right, or in an organism. At that time, these glyoxysomes converts the proteins and lipids into the carbohydrate. Okay, so these are the different organelles present in the eukaryotic cell. Okay, now move to the first topic, that is cell membrane. As we know, the cell membrane, it is its structure is explained by a model that is known as fluid mosaic model, right? That is given by fluid mosaic model, and this fluid mosaic model was explained by two scientists, that is Singer and Nicholson, right? Singer and Nicholson were the two scientists, or you can say the biologists, who explained the structure of the cell membrane, in which they express that this cell membrane is composed of basically two biomolecules. One is lipid and another is protein. The lipid content is 60%, whereas the protein content is 40%. If you can see in this picture, some proteins are present on the surface which can be easily removed from the cell membrane, and some are penetrating throughout the cell membrane which cannot be easily removed. So these are the two different types of protein which are present on the surface. They are helping in the communication. Those are known as external proteins. And those who are getting embedded, properly embedded inside the membrane, those are integral proteins, right? And these integral proteins are helping as a channel in the transport of the molecules. Okay, external proteins are helping in a communication and sensation, whereas the integral proteins are helping as a or working as a channel for the transport of the material. Okay, so here we are having the concept check test for you. The question is, the plasma membrane consists mainly of A, the phospholipids embedded in a protein bilayer, protein embedded in a phospholipid bilayer, C, protein embedded in a polymer of glucose molecule, and protein embedded in a carbohydrate layer. So for this question, I'm giving you next 30 seconds. For the 30 seconds, the poll will be launched, and after 30 seconds, the poll will be removed, right? So here is the poll. You have to give your response. Okay, so I have got the response. Here I am ending the poll, as everyone has already given the response. Yes, that the correct answer is option B, that proteins are embedded in a phospholipid bilayer. As we have seen in this diagram also, these hairpin-like structures are the phospholipid bilayer in which the proteins are embedded. Very good, everyone. So now move to the next structure, that is cell wall, which is present around the plant cell. Okay, so these are the non-living rigid structures, and they can be made up of different components, right? If we talk about algae, they are made up of cellulose, galactans, mannese, minerals such as calcium carbonate. But when we go for the plants, they can be made up of cellulose, hemicellulose, pectin, and protein. If we go to the bacteria, it is made up of peptidoglycan. If we go to the fungi, it is made up of chitin, right? Next, we are having endomembrane system. As I told you, these all four organelles are involved in the formation of membrane. Starting with the ER, ER is having the two different structures, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum are having the ribosomes on their surface, due to which they are helping in the production and modification of protein, right? But when you see the structure of smooth endoplasmic reticulum, in smooth endoplasmic reticulum, no ribosome is present. So they are involved in the synthesis of lipid, right? So as the lipid and protein both are involved in the formation of the cell wall or cell membrane, these both went to the Golgi bodies. Okay, after endoplasmic reticulum, they went to the Golgi body for the packaging and tagging. Packaging and tagging is required because without tagging, we cannot deliver these proteins and the lipids to their specific sites. Then we are having the lysosome. These lysosomes, we can say the secretory vessels produced by the Golgi apparatus. And these lysosomes contains the hydrolytic enzymes, right? These hydrolytic enzymes are helping in the digestion of waste and destruction of foreign element, right? So this is done by the lysosome. But if the cell got damaged, if the cell got damaged, these lysosomes burst. These lysosomes burst and lead to the suicide of the cell. Suicide of the cell. That means they themselves destroy their own cell. That is why these lysosomes are also known as suicidal bags. Okay. Now we are moving towards the next structure. These are the single membrane structure. These are the single membrane structure or a sac, like an empty sac, right? These structure having the outer membrane, which is a single membrane known as tonoplast, and they help in the transport of iron as well as the several materials, right? So in plants, you can see the vacuoles are bigger in size, and in animals they are very smaller in size. Smaller in size with a high number, right? And in plants, bigger size with a very low number. 1 or 2 vacuoles found. One of the kind of vacuole is contractile vacuole, which is present in the amoeba, helping in the excretion of waste. Some there it is helping as a food vacuole, engulfing the food and helping in its phagocytosis or endocytosis, right? The next picture here, we are having the mitochondria. It is a double membrane structure. It is a double membrane structure. The outer membrane is smaller than the inner membrane, right? And inner membrane is present in folds to increase the surface area, for reaction. For all the enzymatic reaction, these mitochondria is having their own DNA and 70S ribosome. Okay, and this is the site for the production of energy. Now we are having next the plastids. Plastids, as I told you, these containing the pigments. They can be colorful, they can be colorless. Depending on that, we are having different types of structures. The chloroplast, which is containing chlorophyll, helping in the formation of food in plants by trapping the light energy, or we can say by the process of photosynthesis. Then we are having the chromoplast, which is a colorful pigment providing color to the flowers and the fruits. Then we are having the leucoplast, as the term itself says, leuco, that is colorless. So these are the plastids which are helping in the storage. They can be of 3 different types depending on the substance they are storing. They can be amyloplast storing starch, alloplast storing oils, and alluroplast, they are present as a storage of protein. Okay. Next, as I have already explained you the ribosome, then cilia and flagella. Okay, the cilia and the flagella. Okay. Next, we are having the centrosome and centriole. Centrosomes are the two perpendicular cylindrical structures. Like these single cylindrical structure is known as centriole. These are made up of microtubules and helping, as you can see here, three circular structures are there. So this is a trimer of microtubule, and they are helping during the cell division for making the spindle fibers and segregating the chromosome from one another. Okay. Then we are having the nucleus. As I have told you, the nucleus was first discovered by the Robert Brown in 1831, and it was done by the staining of the basic dye. The component which are present inside the nucleus is acidic, so they gain the basic dye, and they give the component which is getting the basic dye as chromatin material by scientist Fleming. And one cell is always having a one nucleus. Like if the plant cell you see, the nucleus can be present in the corner because of the larger size of vacuole. And if you see the animal cell, it is present in the center, right? So the cell, this nucleus is present in all the cells except the mammalian RBCs and the sieve tubes of the vascular plant, because sieve tubes are the dead cells. Okay. So these nucleus having the nuclear envelope, that is the nuclear membrane, a fluid material, just like the cell is having cytoplasm, the nucleus is having nucleoplasm. Then the chromatin material, that is the DNA fragment, right? That is the DNA fragment, or we can say the thread, and the nucleus. As you can see, there is a dense smaller body present in the nucleoplasm. Now we are having chromosomes. As the cell keep on dividing, right? When the cell enters in the division phase, at that time, this DNA, which is present, the DNA, which is present in a ribbon-like structure, they start getting condensed, right? They start getting condensed when the cell goes under division. Okay. So with division or during division, this DNA ribbons get condensed and form the chromosomes, right? So these chromosomes are made up of chromatin material. If we talk about the chromatin material, it is a DNA and the histone protein, which are helping in the condensation of DNA. Okay. And if you see the structure of chromosome, there are two different structures. One, as you can see in the middle, these are the centromere. The centromere is having a plate surrounding it, a protein plate surrounding it, which is known as kinetochore, providing the attachment to the spindle fiber, right? Whereas it is having two arms, right? These arms are known as chromatids. The upper one is known as P arm and the lower one is known as Q arm. And depending on the size of these two arms, we are having four different types of chromosomes. They can be metacentric, submetacentric, telocentric, and acrocentric. In all these four chromosomes, if you see with the line, the P arm size keep on reducing. Okay. So as I have told you, we are having four different types of chromosome. Metacentric is having equal P and Q arm. Submetacentric is having slightly smaller P arm as compared to Q. The acrocentric is again having smaller one, and telocentric, in which the P arm is almost invisible because the centromere is almost present at the one edge. Okay. So these are the difference we have studied, that plant cell is having cell wall, animals are not having. Plant cells are also present in plant cell, but it is absent in animal cell. The plant cells are having larger vacuole, whereas the animal cells are also having vacuole, but they are very larger in number and smaller in size. Whereas in plant cells, the centrioles are not present for the cell division, whereas in animal cell, these centrioles are present. Okay, so here we are having a question for you, that select the correct statement from the following regarding the cell membrane. A, the sodium and potassium ion move across the cell membrane by passive transport. Protein make up 60 to 70% of the cell membrane. Lipids are arranged in the bilayer with polar heads outward and inwards. Okay, and the last one is fluid mosaic model of the cell membrane was proposed by the Singhal and the Nicholson. Right. So you have to find out the correct statement. For that, I'm launching the poll, and this poll will stay for next 30 seconds. Okay. So for 30 seconds, the poll will be here. You have to feed your correct answer, your four statements, which out of which you have to find out the correct is like this. A, the sodium and potassium ions move across the cell membrane passively. B, protein makes up the 60 to 70% of the cell membrane. C is saying lipid are arranged as a bilayer where polar heads are present inward and outward. Whereas the D statement is saying fluid mosaic model is proposed by the Singer and Nichols. Okay, so with this, only 5 seconds are left to close the poll. Okay, and with this, I'm ending the poll. As I can see, most of you have answered. So yes, the answer is option D, that the fluid mosaic model was proposed by the Singer and Nicholson, which we have studied in the earlier. The 60-70% of the content of cell wall or cell membrane is lipid. The sodium and potassium ions, these are the ions, that is charged material. So they need active transport instead of passive one. Whereas lipids are arranged in the bilayer where the polar heads are present only outward and the non-polar chains are present inward, right? That is why they are working as hydrophobic as well as hydrophilic molecules. Okay. So next topic we are having, that is cell cycle. So cell cycle, as you know, that is the life cycle of a cell in which we are having the two phases, the interphase and the division phase. The interphase is also known as the preparatory phase. Preparatory phase, right? In which the cell grows and prepare itself for the division. And next we are having the division phase in which the cell divides. It can be mitosis or it can be meiosis. Then we are having the cytokinesis, that is the formation of cell membrane after division of cytoplasm. Okay, so as we know, there are two types of cells, the non-somatic and the somatic one. Somatic cells are those which are not playing role in the formation of, that is the normal cells. Those are the normal cells of body, not playing role in the formation of gametes, whereas non-somatic cells are forming the gametes, right? So if gametes are forming, gametes are having reduced half number of chromosomes. For that, we require to reduce the number of chromosomes during division, right? So depending on the type of cell going under division, it can be mitosis or it can be meiosis. So as we can see in the interphase is composed of three different phases, G1, G2 and S phase. G1 phase is a preparatory phase and metabolically active, right? That means the cell starts increasing its size with the content. S phase, that is synthesis phase, where the duplication of the DNA is taking place, and the third phase is G phase. Because G phase is also known as the protein synthesis phase, as during the separation of the chromosome and during the division of the chromosome cell, we require the protein. So here all the major protein synthesis is taking place in these three phases. and these interface composed of 90% 5% time of a cycle. If we take a human cell, in human cell, complete 24 hours are required for a cell to divide, out of which 21 hours is went under interphase, only 3 hours are required for the metaphase or mitosis or meiosis. Okay, so here we are having some phases, and you can see these M phase or division phase is divided into 4 basic phases, that is prophase, metaphase, anaphase, telophase, which are followed by the cytokinesis. So let's discuss first. The prophase is the phase in which the condensation of the DNA and the formation of the chromosome is taking place. Along with that, the nuclear membrane gets disappeared. Then we are moving towards the next phase, that is the metaphase, in which all the condensed chromosomes get aligned in the middle of the cell, or we can call the plate as a equatorial plate, to which the spindle fibers get attached to the kinetochore. In the anaphase, the spindle fibers have started getting shortening, due to which the chromosomes fold under two opposite poles of the cells. Next is the telophase, that is decondensation of the DNA is taking place, right? Decondensation of the DNA is taking place, and again the nuclear envelope will appear along with the organelles. As these things complete, it is followed by the division of the cytoplasm and the formation. After the division of the cytoplasm will lead to the formation of the two daughter cells. That is how the nuclear division completes. And the same process also takes place in the meiosis. But as I told you, the meiosis is taking place in a gametic cells, where we have to reduce the cell number or chromosome number. This is including extra 5 phases, which are present in prophase 1. Prophase 1 is having the 5 extra phases. That is diplotin, zygotin, pachytin, leptotin, zygotin, pachytin, diplotin, and dikinesis. Okay, in which the zygotin stage is marked by the formation of synaptonymal complex, whereas pachytin is observed as a crossing over formation, right? Where in dikinesis phase, this, or we can say the diplotin phase, that crossing over completes and the recombinant sister chromatids get separated. Then again, the same procedure is followed by the metaphase, anaphase, prophase, telophase. First division completed, this meiosis 1 is known as reductional division, because in this metaphase and anaphase only the chromosome number gets half. Then the meiosis 2 starts, which is identical to the mitosis. And this mitosis is again composed of, like as I told you, the 4 stages, that is prophase, metaphase, anaphase, telophase, and last is cytokinesis. If you see these cell division, the mitosis is leading to the formation of 2 cells at the end having the equal number of chromosomes. Then we are having meiosis leading to the formation of 4 number of daughter cells with a reduced chromosomes, right? So with this we have completed our cell unit containing 2 different chapters, that is cell, the unit of life, and the cell division and the cycle. Okay, so in the next class we'll start with a new chapter, and that is all for today. If you guys are having any doubt, you can leave your doubts in the chat box, so that we can resolve it in the next class. And before leaving the class, you have to give your feedback to this NPS poll. So here the NPS poll is launched, right? So give your feedback, and after giving your feedback, you can leave the meeting. Okay, so thank you everyone for giving your feedback. Here I am ending the NPS poll, and with this we are ending our class. We'll meet tomorrow. We'll meet the next weekend with a new chapter. Okay.